make it open and accessible for people that are entering the field from other areas. So we're very grateful to have people here that have a diversity of skills beyond molecular biology. So if your background is in computational biology or microscopy and plant development, we want to make sure that everybody understands the basic details of how to design a circuit, since that's for the goal of our hackathon today. So I'm going to give the first part of this, and then afterwards Sarah's going to talk about a more advanced circuits. So we are going to start, or our learning goals for this session basically is to think about exactly what a construct looks like, which parts of that construct we have to design and which parts we just choose. We're going to think about those plasmid backbones and how to select them. And then we're just going to recap some of the really simple constructs that are used to engineer plants and how they work. And start talking about how we can manipulate gene expression in plants. So starting right from the basics, since what we're going to be doing today is designing genetic circuits, which are ultimately going to go into some kind of a plasmid and hopefully into make some engineered plants. We're just going to talk a little bit about those plasmids, which are bacterial. So these can be very simple cloning plasmids, such as the one on the left-hand side of this slide, which may only contain just a really few basic features. So minimally, these will have an origin of replication to enable those plasmids to replicate in the bacterial host cell. They're probably also going to have a selection marker required for the maintenance of that plasmid by the host cell, and that's most often going to be a antibiotic resistance gene, but it could also be something else. And most commonly, or often, but not always, there may be a specific cloning site where the new genetic material, or the material that you're going to be focused on designing today, is going to be added. And cloning into that site may either disrupt or replace a cloning selection gene that enables the easy identification of clones in which your inserts have been successfully inserted. But transformation of plants is often, but not always, achieved using the bacterial vector Agrobacterium, often Agrobacterium tumefaciens, to transfer the genes of interest into plant nuclei so that they can be integrated into the genome. And so these plasmids, or the plasmids used to use that transformation system, are usually binary plasmids, and they have two origins of replication. They also have an origin of replication for E. coli, because that's where we still do our cloning, but they'll also have an origin of replication so that that completed plasmid can then be transferred and grow in the Agrobacterium cell, which will recognize a different ori. There's also a bacterial selectable marker still. So there's many features that will overlap. So they'll also often have the cloning site where we insert our DNA. But in these plasmids, the region that's going to form what we call that transfer DNA, that tDNA, that's going to be transferred into the plant cell via the Agrobacterium's type 4 secretory system, aided by virulence proteins that are encoded by the Agrobacterium that are going to protect that transfer DNA from nucleases and enabled it to pass through the plant cell and enter the plant nucleus and ultimately be inserted into the genome. So that region is going to be flanked by two sequences that we call the border sequences, the left and right border sequences. And outside that right border, there will be a sequence called the overdrive, to which virulence protein C binds to increase the production of those tDNA strands. So as I said, all that new genetic material that you will be designing and building will then be inserted between those left and right borders. And so some plasmids may already contain, as well as just this cloning site, some of them will already contain a selectable marker gene that allows the selection of transgenic plant cells to be distinguished from those that haven't had the tDNA inserted into them. Okay, so how do we choose what plasmid to use? So the first selection criteria is obviously going to be how we intend to deliver the DNA to our plant cell. So if we're going to choose to deliver by agrobacterium, then we do need to select one of those binary constructs, or at least plasmids that have those features to allow transfer into the plant cell by that bacterial chassis. Alternatively, if we're going to be not going to be using that, but we're going to be transferring either directly into plant protoplasts, or we're going to be delivering that DNA directly for biolistic delivery, we're not going to replicate that plasmid in an agrobacterium cell. And often for both of those methods, you really use quite a lot of DNA, maybe can be up to 20, 30 micrograms of DNA for some of those methods that you need. So it's better to just do this, keep this in Nicola and keep it as a very high copy plasmid. And because of this, smaller plasmids will make your life easier. So it's better to avoid the binary backbones, which can be a little larger, and to keep it into a smaller backbone plasmid. So other things that we might select as well for when we're selecting suites of plasmids that we like to work with, we might have particular cloning sites that work for the things we want to build. So common things that are commonly used in plant molecular biology labs are things like the gateway system and Golden Gate system, and we'll talk a little bit more about some of those when we get to the build section. And then there may be other features of your plasmid that are already in those plasmids, such as those selectable markers, such as plasmids that are specifically tailored for protein production, that might be useful. So let's turn our attention now completely to the section of DNA that's going to be functional inside the plant cell. And we're going to do this by, first of all, looking at the very simplest synthetic genes. So people have been building these really simple synthetic chimeric genes since the 1980s and inserting them into plants. And the very simplest thing to do with any gene is basically to overexpress it. And so what overexpression means is it means that that gene of interest is going to be expressed at a high level in most or all of the cells in that organism. So that means we're going to use a strong constitutive promoter to express our gene of interest. So this might be done transiently. So it might just be in the cells to which your construct or your tDNA has been delivered, in which case there's no need for a plant selection gene. Or it might be expressed throughout a transgenic plant, in which case we generally also need a selectable marker gene to enable the selection of transformed cells and the regeneration of a transgenic plant. So an overexpression plasmid or an overexpression cassette, it may just have this one gene or with your gene of interest, or it may have your gene of interest plus your selectable marker cassette. And as I said, so your overexpression construct is generally going to consist of a strong constitutive promoter, and the one that's most often used, at least for dicotyledonous in this plant, is the cauliflower mosaic virus 35S promoter, your gene of interest coding sequence, and terminator sequence. But if we want to do more complicated things, one of the things that we will often consider is all the ways in which we may wish to control the expression of our genes or our genes of interest. So in expressing a new gene, we might want to control where, when, how much a gene is expressed. And this is important for manipulating plant growth and development. But even if our aim is, for example, to maximize the yield of a new natural product, we might wish to limit expression to a time or a particular tissue to avoid interference with normal growth and development. It can also be useful to make expression contingent on a particular condition, or if it might be useful if expression responds to environmental changes such as heat or light. And it's also useful for the user to be able to retain control over the expression by making it dependent on the presence of a specific molecule that's not normally present in that plant or its environment. So other things we can think about is once that gene has been activated, do we want that expression to stay on forever, or should it be possible to turn it off, which will require a switch? should expression levels be activated periodically or cyclically? For example, every day, every spring. We might want to think about how quickly we want activation to happen. Should it turn on slowly and reach maximum production immediately, or do we want it to ramp up? And then we can also consider natural processes in plants, such as response to temperature, like temperature-dependent flowering, circadian rhythms, and think about the types of control important for these. The amount of the protein, which can be our gene of interest in a cell, is controlled at multiple levels. So it can be controlled at transcription. It could be controlled at the post-transcriptional level by the stability, for example, of that messenger RNA, or by gene silencing. It can be controlled at the translational level by the codon usage and the efficiency of translation, and by post-translational modifications. But most of the time, because synthetic circuits are initiated at transcription, we generally focus on transcriptional control. But there are tools available for you as well to be able to think about manipulating gene expression of your gene of interest, or genes in the plant at all kinds of level. So let's just refresh our knowledge really briefly about gene structure and the role of regulatory sequences in transcription. So these regulatory sequences all contribute to gene expression. So these promoter and enhancer regions regulate transcription of the gene into a pre-mRNA, which includes exons, which are shown in red, and introns shown in gray, between the 5' and 3' untranslated regions. And then that pre-mRNA is modified to remove those introns. A 5' cap and poly-A tail are added, unless it's a small RNA. And then the 5' and 3' regions regulate translation into protein eventually. Okay, so transcription factors bind to the promoter and enhancer regions of the DNA, and they interact with other transcription factors and mediator proteins and recruit that RNA polymerase complex. Regions of the DNA that are distal to the transcriptional start site can be brought closer through the interaction of transcription factors and mediators. It's kind of a looping effect. The ability of transcription factors to access DNA and recruit RNA polymerases is in fact affected by a number of different processes, including alterations chromatin structure, modifications to DNA and histones, and interactions between those distal regulatory elements and promoters. So all of these things can affect how a given regulatory region is going to behave in the cell. So if we don't want to constitutively express, we can go for a tissue-specific expression. And the way that this is most often achieved is by fusing the promoter of a plant gene to our gene of interest. Now, sometimes we know the expression profile of that plant gene, but often plant promoters are not well characterized. So some of the problems that we run into is identifying promoters, deciding how long it should be, if it has distal or trans cis elements. All of these things can mean that when we can select the promoter of a gene that we know its expression pattern of, but it might behave differently in a new genomic location, and it might behave differently in a synthetic cassette. So there are various bioinformatic analyses that we can use to predict these, and chromatin accessibility information can help. But that we almost always do generally need to test those. And this is most often done by fusing these to a reporter and visualizing this expression pattern. So on this slide, we can just see the promoter region of the Arabidopsis PIN1 gene fused to the coding sequence of PIN1 fused to GFP. And this is giving us the expected expression pattern in the green, and the red is just propydium iodide staining. So coming back as well to the deluge of data now that's emerging from single-cell transcriptomics, expression patterns of things like PIN1 are very well studied and have been for many years. So I just want to show you a new data from a single-cell transcriptomics being used to identify genes in a particular cell type. So in this study, the authors used single-cell transcriptomics to identify a cluster of genes for companion cells. We know very little about control of expression there. And they selected genes that are unique to this cluster, which is shown here in orange. And fuse that to the Venus reporter, and you can see in the longitudinal and cross-section and lateral cross-section that it does indeed have companion-specific cell expression. So the very last thing I'm going to talk about before passing on to Sarah to talk about more complex circuits is kind of the basics. The basis of many circuits in synthetic biology is to have an inducible promoter. And here I'm talking about those that are inducible by an external chemical signal. So these really, we talk about them as inducible promoters, but they really consist of two elements. They consist of this synthetic transcription factor that then binds to a cognate synthetic promoter. And usually then it will bind to specific operators or binding sites just upstream of the transcriptional start site. So you fuse your synthetic promoter to your gene of interest, and then you co-express your synthetic transcription factor. And then in the presence of the signal, it's able to bind to the operator. So these synthetic transcription factors, or these inducible synthetic transcription factors, often have three domains. They often have a DNA binding domain, which is what's recognized and binds to this binding site or operator. They have the effector domain, which is the thing that either activates or represses expression. And then they'll have a ligand binding domain, which will often do one of two things. It will either, in the presence of the ligand, it will induce a conformational change that will allow it to bind to DNA, or sometimes that ligand will be, or that ligand binding domain will hold it in the cytosol unless the ligand is present, and then that will allow it to dissociate and be translocated to the nucleus. So there are other systems available. We have somebody in the audience here that has more experience of optogenetics than I probably do as well, but light has been used for controlling gene expression in plants. I'm not going to go into this in detail. I've just put it here as an example of the other ways that we have now of being able to externally control gene expression in plants. And I also want to just briefly note that you can also, as well as being able to have inducible expression by external chemical signals, you can also use internal chemical signals. So one example of this is the auxin responsive promoter, and this is a synthetic promoter similar to the ones that I just described, but instead of having binding sites for a synthetic transcription factor, it has binding sites for ARF proteins or auxin responsive transcription factors. And these exist in plant cells, and they activate expression in the presence of auxin. So one of these well-known promoters is known as the DR5 version 2 promoter, and it has 9 copies of the ARF binding site, each one being itself being an inverted repeat. And we can see that when it's inserted into the plant genome, it's most strongly expressed in the cells in which auxin accumulates, and there's not really very much expression in cells from which auxin is known to be transported away. So I am going to finish there and leave Sarah to talk to you about how all of these elements can be used to construct more complicated circuits. So hopefully we've just now covered the basics of how to, where we put our synthetic DNA, you know, something about plasmid backbones, and you know, something about some of the most basic expression constructs or synthetic cassettes for overexpressing, for manipulating expression, in a tissue-specific or an inducible manner.